Starts, I just want to introduce myself really quickly. My name is Shivani Avila. I'm here representing the Carolina Center for Public Service, and I'm going to be serving as your point person for this event. So if you have any sort of technical questions or need assistance, just feel free to reach out in the chat. I'm also here to help with any questions about CCPS programs or Carolina Engagement Week. At the end of the event, we're going to have a brief evaluation survey. It should take no more than 2 to 3 minutes. Your feedback is really helpful to us so that we can make sure that these events stay being relevant and are great for you all, our prime audience. And so stick around for that. I'll put the link at the end. And if anyone here needs Campus Life Experience credit, I can also put that QR code at the end of the event. So thank you so much. And I'm going to go ahead and hand it off to Marianne and Julie.
Thank you so much. Well, we're very excited to be here. My name is Dr. Julie Horvath, and I'm a senior research scientist at the Renaissance Computing Institute at UNC Chapel Hill. And we are going to be talking about one of our projects. And so we have a good mix of people who are students, potentially educators, and other researchers. And so we're going to try to hit kind of the broad breadth of our project because it spans multiple different angles. If you have questions, you can put them in chat, but we are going to ask you to participate a little bit. So please be ready to pop some stuff in chat. We'd love to get you guys engaged. So as I mentioned, I'm Dr. Julie Horvath. We're going to be talking about Monkey Health Explorer because it's a project that we have in North Carolina and it now is across Puerto Rico as well. Our collaborator Francelia is here, so thank you for joining, Francelia. And we're going to talk about this project and engage in, how to engage students and how to do health-related research with this project. So if you have connection questions, if you have any interest, please connect with us and let us know. And I am going to talk a little bit more about this in a minute, but I want to first let Marianne introduce herself, and then I will take it away and start talking about the project. So Marianne, why don't you go ahead and introduce yourself?
Hi, I'm Dr. Marianne Barrier. I am the lab manager of the Genomics and Microbiology Research Lab here at the Museum of Natural Sciences in Raleigh. And I've been a part of this Monkey Health Explorer project doing collaboration with Dr. Horvath for several years now. So excited to get to share this with you.
Awesome, thank you. All right. So as I mentioned, I'm at the Renaissance Computing Institute. Marianne is at the museum, and this is a partnership between our institutions, and it's a collaboration that's been running for many years, and hopefully for many more. So Marianne, if you can go to the next slide. Today, I'm going to kind of go over some of the goals of what we want to talk to you about. I'm going to give you a little bit of background, tell you why should you care about this project, why is it relevant to your daily life, your health, etc., maybe your career as well. And then Marianne can introduce the Monkey Health Explorer project itself. So if you want to engage and participate in the project, we're going to tell you how to do that. Then we'll give you a little bit of lessons learned, how to connect, how to get involved, and maybe some future directions that we have as well. All right, so what I ask you, so here's your first participation problem, is we'd like you to introduce yourself in the chat. So tell us your name, tell us, are you a student, educator, researcher, what your role is, tell us your institution. I imagine there are going to be a lot at UNC, but if you're not at UNC, that's awesome. So please let us know. But the other piece that we really want you to tell us is what is your favorite primate? So we're going to talk mostly about rhesus macaques today, but tell us what's your favorite primate. So please do that in the chat and we will kind of monitor that and see where you guys are. And then I'll be talking throughout that. Chimpanzees, yes. All right, so Marianne, if you can go to the next slide. So I wanted to give you a little background about how this project really started. It started when I was young because I grew up in Michigan. If anybody's from Michigan, I grew up right here in Michigan. Spent a lot of time in northern Wisconsin on a lake similar to what you see here. So I had this little boat. It was also red, had a blue seat. And I spent a lot of time outdoors because I love the outdoors. But I quickly learned that there were some animals I could get really close to. So the chipmunks and the chickadees, I could tame them. They would eat out of my hand. Probably not that smart, but I was young. I loved animals. But I also noticed that the squirrels, the crows, they wouldn't get near me. So it was really hard to tame them. And I got really captivated by, what controls animal behavior? Why are some animals easier to tame or, more amenable to humans and others are not? And so that kind of drove some of my research experience. So when I went to college, I went to Michigan State University, and I explored different career opportunities. So I thought I wanted to study animal behavior. And so that's what I started out in. All my classes were focused on animal behavior. And I quickly learned that that wasn't quite the right path for me. So I started taking some genetics classes and I fell in love with genetics. I was fascinated by how one base pair change in DNA could actually, cause a disease in one person or animal. And, that one base pair change, a different base pair meant somebody was healthy and happy. And so that fascinated me. So I changed from animal behavior to genetics. I studied Nubian goats, as you see here, and Solares cattle, and we studied a genetic disease in them. But that got me really interested in this animal health piece, but I still was interested in the behavior. So I wanted to find a way to connect with the behavior. Again, I loved animals, and so I worked at the zoo. So here's me with Julie the camel. Julie, me plus Julie the camel. So lots of fun. So got me really interested in animals, and that kind of is going to be the thread through the talk today. So Marianne, if you can go to the next slide. So my favorite primates are lemurs. My very, very favorite one is probably the blue-eyed black lemur. And you can see an example of a female blue-eyed black lemur who is not black at the bottom. The female blue-eyed black lemurs are the dominant ones. And so the animals were named for the male counterpart, but it's actually the blue-eyed black lemur female that is more captivating to me. So here's an example of her picture. And if you go to the Duke Lemur Center, it's a great opportunity to meet all these different animals. It looks like we have a really great mix of people's favorite animals, favorite primates. So we have capuchins, lemurs. Thank you for the lemurs. Shout out to the lemurs. Orangutans, spider monkeys, chimps. Yes, lots of chimp love. That's very common. Some macaques. Awesome. Francelia, I'm guessing maybe you typed in macaque. Awesome. Proboscis, tarsiers. Yeah, awesome. So great. Thank you for joining and participating in that. That's lots of fun. We're not going to talk too much about lemurs anymore, but that kind of shaped some of my research. So I worked at Duke for a while, but then I got this opportunity to work at the Museum of Natural Sciences. And so here's the lab that I was the head of, the Genomics and Microbiology Research Lab. And you see it has all glass walls, and we had some primate skulls in the window, a really fun way to engage the public, because the public can walk up to this research lab and watch science in action. So even though they don't get to go in every day, you can still see what's happening. And so it's different than an academic setting because it's a great way to engage the public. And so if you come out of the lab, go to the bathroom, go grab lunch, and then there's the public there, and you can kind of talk to them. And so a lot of what we do is find ways to engage the public. And so that's why this project has become so successful, because we've really engaged a lot of the public locally and around the world. So Marianne still works in this lab as the lab manager, and we have now this partnership between RENC and the museum. And so we have an Illuminating AI talk series and lots of other opportunities. So if you're engaged and interested in the museum more, certainly message us and we'll find other ways to connect. But that's kind of the driver of some of the research that we'll be talking about, kind of stemmed from the museum. All right, so on the next slide, I want to call out some of our great collaborators, because as I was working in this museum space, we were finding ways, how do you engage the public in primates? You can't have primates in this lab, and so you can't have people study the primates themselves in the lab, but we were looking for ways to engage the public. And so this project that we'll be talking about is one way that we did that. And so Francelia here is one of our collaborators who's really taken this on and taken this project to Puerto Rico, because like I said, we started in North Carolina. She's really taken it to Puerto Rico now. Dave Gandhi is one of our grad students at UNC, and he's been working on some of the data analysis pieces, which has been super helpful. We have collaborators at UPenn, Caribbean Primate Research Center, University of Exeter, Arizona State. It's actually a huge collaboration. There are more than 50 people involved. These are some of the ones that started out on this project and are closer collaborators with us. So Lauren, for example, studies behavior. And so some of the behavioral information I'm going to show you is actually some of Lauren's work. So on the next slide, you can kind of see an example of some of the macaques that we study. So here they are just out in the field, free ranging, and there are people who are studying them. So recording focal follows of, what are the animals doing? Are they interacting? Are they grooming? Are they eating, etc. So again, a really great opportunity to study these macaques. And so on the next slide, I'll kind of highlight that these macaques are really useful because they're some of our closest relatives. They are not our very closest relatives. Those are the chimpanzees. But the macaques from Asia and Africa, other other animals from African and Asian origins are really somewhat close to us. So we maybe shared a common ancestor 25 million years ago. So it sounds like a long time, but that means at the DNA level, we're more than 90% identical. And so a lot of our genes are very similar and have similar functions. And so that makes macaques a really good model for studying some of the things that we're going to talk about today. So they're great biomedical models. There are a lot of macaques across primate centers, and as you'll hear, across parts of Puerto Rico. They're used to study HIV, alcohol research, Alzheimer's, vaccine research, and behavioral research. And so a lot of this information is really valuable and important to understand humans. And so that's where the macaques come in and why you should care, because these are really relevant to a lot of information about you and your health. So if you can advance it again, Marianne. So one fun fact I wanted to add here is that the Rh factor, if anybody knows if you're Rh positive or negative, that Rh factor was identified in rhesus macaques by studying the macaque blood. That's why it's got that Rh factor. And the Rh factor is a protein on the surface of red blood cells. And so if you're Rh negative, you don't have that protein. If you're Rh positive, you do have it. The majority of people are Rh positive, but it's really important if you are a pregnant woman and you have a child who's Rh positive and you're Rh negative, that creates some implications for you and your health. So again, really important to understand some of this, and the macaques are really useful as models to help us understand a lot of this. So on the next slide, I'm going to talk a little bit about macaques and give you some more insight into them. So they are found across parts of India and China. This is their formal name. And even though they're in India and China, the population that we study is in Puerto Rico. And so on the next, if you could advance it one more, we study them on this really small island called Cayo Santiago, which is off the coast of Puerto Rico. And there are more than 1,000 macaques that live on this really small island. They were all brought there in the 1930s by Clarence Carpenter. He brought about 400 animals, and now that has expanded to over 1,000, like I said. And they're all Indian origin monkeys. And they're semi-free ranging. So they do, they don't have cages on the island. They do semi-free range and they move around, but it's not a very big island. So they do get provisioned with monkey chow. So a lot of the Caribbean Primate Research Center will provide them monkey chow and water. And so that keeps them, surviving and healthy. And then we can study their behavior and study other aspects of their health. And so we're going to be talking a lot about the health of these monkeys. So again, why should you care? Well, these macaques are great models for health and aging, and we started studying out their health and have really advanced to connect with aging and studying aging more broadly in these macaques. And so we're studying the long-term behavior of some of the monkeys, as well as looking at the health, but we're now starting to correlate with aspects of genetics. We're looking at environmental effects. And so one of the projects I'm going to talk about that we recently started and published on is really looking at some of these environmental effects and how that impacts your health. So if anybody is aware, in 2017, Hurricane Maria hit Cayo Santiago and Puerto Rico, and it was a really large and dramatic storm, and it hit this island. You see on the left is a picture of Cayo Santiago before Hurricane Maria, and on the right is a picture after Hurricane Maria. And so you see the deforestation. It used to be very lush and green, and it looks very different now. And the monkeys utilized that shade and would spread out more and use the shade as a resource. And now that this hurricane went through and there's a lot less shade, the monkeys have to actually change their behavior a bit, and they have to get closer together to utilize some of the shade because there's less shade on the island. And so that's changing some of how the monkeys behave. But this was also a really dramatic impact on the monkeys. There was a lot of stress. As anybody's aware, when we get hurricanes in North Carolina, there's a lot of stress on humans. And so just imagine if you were living outside as these monkeys were, how did they deal with some of the hurricane aftermath? So one of the things that our colleagues and I did is we actually wanted to look at the monkeys before, monkeys that weren't exposed to the hurricane, and compare that to monkeys that were exposed to the hurricane. So before and after, essentially. And so on the next slide, I'm going to highlight some of what we found. So if you study any animal, as any animal ages, different genes get turned on and off. So different genes are expressed at different time points in your process of aging. And as you go through chronological age, you're 1, 2, 3 years old, that's your chronological age. But your biological age sometimes changes differently. So if you have a stressor, or if you're smoking or eating some food that maybe is damaging your DNA, sometimes there are parts of your body, or maybe your whole body, that is going to age at a different different level than your chronological age. So we call that your biological age. If different genes are turned on and off, that's looking at your biological age. And so what we did is we could look at monkeys that had not been exposed to the hurricane. We looked at their blood and we looked at which genes were turned on in their blood or turned off in their blood. And we compared that to monkeys who had gone through that hurricane and looked at which genes were turned on in the monkeys that had been exposed to the hurricane. And essentially the hurricane actually induced immune system gene changes and inflammation changes. And so that hurricane, that major stressor, actually changed which genes were turned on in these monkeys. And what that meant for aging, if you advance next, Marianne, the animals that went through this hurricane and experienced it actually had advanced their biological age. So their gene expression profiles looked different because they had gone through this traumatic event. And so it was equivalent to almost 2 years of aging in these monkeys, which would be equivalent to about 7 to 8 years in humans. So this dramatic environmental effect really impacted the monkeys in which genes were turned on and off. And that's really relevant to humans because we're probably seeing something similar if we go through some traumatic events like a hurricane. So if you can advance again. So one of the things that we then wanted to do was say, okay, we're seeing some of these dramatic effects. Can we now start looking at health of these monkeys? And can we actually start looking at the blood of the monkeys to learn a little bit more about their health and how the aging process maybe connects to their health? So this leads us to some of our scientific questions that we'll outline today. And one of them is, what can we learn about monkey health if we start looking at their blood or look at blood smears? And then how does monkey health connect to things like behavior or genetics, et cetera? And so we're starting to make some more of these connections. And again, the research team that we have is really broad, and it's kind of this team science approach because everybody has a different background, and we're bringing it all together to answer some of these larger questions. So yeah, you can advance again. And so the project we're talking about is called Monkey Health Explorer. It's on this people-powered research platform called Zooniverse. So type in the chat, has anybody ever used Zooniverse? So you can just type yes or no. Have you ever used Zooniverse? Have you gone on there and participated in one of these projects? There are more than 70 projects on Zooniverse, and Monkey Health Explorer is one. I see a lot of no's in the chat, so that's awesome. This is a great place for you to go and participate. And if you're interested in careers and finding out what kind of careers you like, this is a great place to go because you can find out and participate in some of these research projects. These are all authentic research projects. And you can find out, what do you like or not like? And then maybe that'll give you an idea of kind of your career path or some of the things you might like to partner with. And so a lot of these are what we call participatory science projects or citizen science projects, which are these partnerships between professional researchers and the public and students to really address some of these questions. And so, like I said, we have some questions about monkey health, and we're going to ask the public to help participate in these projects and give us some insight into the monkeys that we can then analyze in the future. And so you can go to this zooniverse.org and find Monkey Health Explorer. You can also find this project on SciStarter, which is another site that has even more projects, but Zooniverse actually hosts our Monkey Health Explorer project. So on the next slide, I'm going to run through a little bit about this project. So here's what the landing page looks like. You can go to monkeyhealthexplorer.org, or you can go there through Zooniverse. And this landing page just shows you, okay, we're studying these macaques. A lot of our collaborators study the behavior, but we're actually getting blood samples. So once a year, we collect blood samples from a subset of the monkeys. It's generally around 100 to 150 monkeys a year. And then we create these blood smear images, which I'll show you in a minute. We now have almost 24,000 volunteers, so you can all be volunteers. I welcome you to this project. We've done more than 2.7 million classifications of white blood cells. And the beauty of some of these participatory science projects is that we get multiple people to look at the same image, and then we look at the consensus. So we make sure that enough people agree on what the consensus is when identifying a cell. And so if there's agreement, then we say, okay, that's a pretty good predictor. We're very confident in what this cell identification is. If we get a lot of disagreement, then that means we flag it for us and we go and look at it. So it gives us a lot of confidence in our results. So on the next slide, I'll run through a little bit about some of the goals and how we go through this project. So as I mentioned, our research goals are, can we look at whole blood from these macaques? Can we look at gene expression profiles? Can we look at the health of the monkeys? Can we also look at their behavior and make some of these broader connections? And so today we're just going to be focusing on the monkey health from the blood, but just think that this is a bigger project that has a lot more facets to it. And the training goal for this is, can we train you to identify some of these cell types in blood? And so if you go to this project, there's a training profile, so you can go through that and get trained. And then as you participate in the project, you actually get a lot better. So Marianne and I weren't trained at the outset. Marianne has done tens of thousands of images probably compared to mine. And so she's great at this. So she has kind of self-taught herself, which is phenomenal. So you can go do that too if you're really interested. So like I said, you collect whole blood, and then what we do is we take drops of that blood on one slide, and we take another glass slide and kind of smear it out to make this blood smear. We've done this for hundreds of monkeys, and then after we have this blood smear, we can stain that smear to identify some of the cell types better. And so you can kind of see it gets stained purple. And then we take a scanner, we send it to collaborators in St. Louis, and they scan this image, and then they send us back a really large scanned image of this blood smear. You can see those two arrows there are pointing to white blood cells among the sea of red blood cells, and the red blood cells are those smaller donut-shaped items in this. And we have now thousands and thousands of images from hundreds of monkeys. And so that's why we need to enlist everybody here, as well as others, to really participate and help us identify what some of these cells are. And so I'm going to turn it over to Marianne to walk through some of this. And I know my side wasn't as interactive, but she's going to have more interaction. So I encourage you to participate and make some guesses. And as you go, we'll learn a little bit more about this project. Thanks. I'll hand it over to you, Marianne.
Great. Thanks, Julie. That was a great introduction to the project and to these monkeys that we're working on. So I'm going to show you, basically teach you a little bit about how to identify these cells and go through this a little bit like how I do this one. I sometimes take this into seventh grade classes as part of the SciMatch program. And so I'm going to first do our little activity. So I don't know if anyone got a chance who may have got the email to bring a little baggie and a couple different kinds of candy or other things of 3 different sizes. So these are the ones that we use in the project. We have Nerds for our small candy, we have Red Hots for our medium-sized candy, and we have Skittles for our large. And so if you have your bag, or you can do this later if you didn't get a chance to gather today, but we're going to make a candy blood smear. So what you do is you take your little baggie. So I've already measured out my small candy, which is 1 teaspoon. So I've got a teaspoon of Nerds in there. And then I'm going to add my medium-sized candy, which is 2 tablespoons of Red Hots. I'm going to add those in there as well. And then finally, I've got my teaspoon of my large size, which is my Skittles. And the reason we use something like the Skittles is that it has multiple colors. And ideally, if you find a candy that has 5 different kind of colors, that way you represent the 5 types of white blood cells. So we put them in the baggie and we make sure it's good and closed. And then we shake it up. And then here's where you get to be a cell tech, and you lay the bag down on your surface. I'm going to show you on my hand, and you just spread the cells across on the bag so that you're smearing them out like making a blood smear. So this is basically to represent like the picture we see here on the screen, which is a little crop of a small section of a blood smear. So just like in our little sample bag and candy, or you can also use beads or other 3 sizes of things. So in that, you can look for your small shapes, your medium shapes, and your large shapes. So on the picture here, you see there's small cells, medium cells, and large. So here's a little way, see if you can help me identify what's in the picture here. So first off, can anyone tell me what the little shapes are? So you can just type it in the chat if you have an idea of the different types of cells that we have in our blood. So come on, someone give me a guess here. They're little bitty ones. I'll give you a hint. They're very important if you get a little paper cut or scrape or something like that. So yes, there we see platelets. So that's what we've got here. So that's our smallest candy. It represents our platelets. For me, it's the Nerds. And then the next size is our medium size. And I've highlighted a couple here, but that's all these big discs in here. So for my candies are Red Hots, represent this part 1 type of cell. So who can tell me what type of cell that might be? And the name of the candy I chose was purposeful in the type to fit with what it is. So these cells are very important. And Yes, red blood cells and bringing our oxygen into our bodies. So we've got that, red blood cells. And then, of course, we've got these two large ones here. What would they represent? It's our final one. I like that we have someone tried a specific example of a basophil. So these are actually two types of white blood cells, and you're going to learn more about how to identify them. The one on the left is called an eosinophil, and the one on the right is a neutrophil. We're going to learn more of those specifics in just a minute. So great job for speaking up, those who identified the cell types correctly there. So that's the start. We look at the cells here. So whenever we go to the doctor, get our blood drawn, and they do this complete blood count where they actually count the cells in the blood, you want to count at least 100 white blood cells. So then you also count, as you're counting, just counting across the smear, you also count the red blood cells and the platelets. And that total amount, they're looking to see the relative amounts of the different types in order to see, it gives an idea about the health of the individual. So these are some numbers just for the average human adult. So you have an idea of how many red blood cells, white blood cells, and platelets we have in a liter of blood. And then the typical ratio of white blood cells to red blood cells is about 1 to 600. So that's why there's so many red blood cells and just a few white in the picture there. So in my class, again, when I take this, I like to show this is, why do we care? Why do we want to look at these numbers? Why do we want to look at the blood? Well, it's to identify, the health of whatever individual. And here I use this as an example. Let's say that the monkey in the middle there looks like maybe they're not feeling so great. And I don't know about you, but I know when I'm sick, I sometimes get a little grumpy, don't quite behave the way I normally do. So this is where we might see a connection between behavior and health, where possibly illness or something may affect the behavior of the monkey. So for the class, when I do this, I say, okay, we're going to look at the blood of this particular monkey and see if we see anything out of the ordinary in the blood numbers that might give us an idea of what's going on. So we have this, and this is something that is called a field guide that's available on our Zooniverse project, so that there's also many, many more example images, but this is like a quick one that gives you the 5 types and some example pictures of each type to help you see the variation, because unfortunately, they're not like a cookie cutter. They're not exactly all the same. And you can kind of see that from these examples. But that's okay. You can still figure this out. If you like pattern matching games, this is the project for you. That's why I enjoy doing this project, because that is very much one of my things that I like. So we're going to go through some of these. I'm going to show you what to look for, and then you're going to identify a few cells here. So right here, we've got the 5-pack of cells where we've got some white blood cells. The white blood cells are the big ones in the middle, and they're surrounded by a bunch of red blood cells. So you can see that, and then there's a platelet right there. And then, of course, the ones in the middle are the 5 different white blood cell types. So I'm going to go through these a little bit at a time and tell you what to look for, and then see if you can identify these. And the numbers that I show as far as how common they are is based on human numbers, because we're still developing the normals for rhesus macaque monkeys, particularly for our population. So here I'm going to show you neutrophils and eosinophils together, because they sometimes can be mistaken for one another. The reason is they're both similar size, and they both have a similar kind of structure to the nucleus, but there are some differences. So first off, just to tell you a little bit about neutrophils, neutrophils are our most common white blood cell. They're 40 to 60% of our white blood cells, and they're our first defense. They fight bacteria and fungal infections, so they're very important in keeping us healthy. And then eosinophils are one of our less common white blood cell types. They're only about 1 to 4% of them typically, but they have a different job. They fight parasites, and have also been shown to fight some cancer cells, and are part of our allergic response in asthma. You may have heard eosinophils mentioned in asthma medication or allergy medication commercials. I hear that pop up every once in a while. So now that we're looking at the 2 of them together, and I did some little cartoony drawings of them to help you really see the distinctions of the difference and similarities between them. So notice that they both are, when you look in the pictures with the cells, they're about 2 to 3 times as big as the red blood cells. So they definitely stand out compared to the reds. And then they both have multiple dark shapes kind of connected by thinner connectors in the center. And so can anyone tell me what that dark shape is. So what portion or component of the cell would that dark shape represent? So you want to have a guess at that? And I'll say that when we're looking at the comparison of these two types, we're looking at that shape as well as the background, which would be the cytoplasm. So the cytoplasm is like what we see surrounding that dark shape. I'm seeing if anyone happens to take a guess. This is something that contains the DNA inside the cell. Yes, the nucleus. So these are the nuclei that we're seeing in the cells, are these dark shapes. So the neutrophils have several of these small lobes, which are these little blobs essentially connected by skinnier connectors, whereas eosinophils usually only have 2 or 3, and they're a little bit larger. And the difference, that's one of the big differences. And the other one is that the background of the neutrophil is lighter. Their granules are smaller and paler than the ones in the eosinophil, which tend to be a little more of a reddish purple. And one of the ways I like to tell people to look for this is compare it to the red blood cells nearby. If the background is similar in color to the inside of the donut, the lighter color, it's more likely a neutrophil. If it's more like the outer ring of the donut, that would be like the darker background of eosinophil. So that's one of the ways to kind of what to look for here. So now that you've got that little quick course in those two cells, tell me which number or which letter here is an eosinophil. So go ahead and type it in the chat. So we've got A, B, C, D. I'm seeing some answers. Very good. See, you all are already experts. Doing excellent here. Yes, B is an eosinophil. You see those two nice large lobes. They kind of look like a pair of lungs. And so that is definitely an eosinophil. Which ones are our neutrophils? So let's switch from eosinophil to neutrophil. C and A. There's actually 2 of them. Both A and D. Yeah. So both A and D are neutrophils. Now, so someone put a C. Now, C is actually a little bit misleading. Can anyone tell me? C is not a white blood cell. Sometimes we may get a shape like that looks, some people may think is, but it's actually something else that we looked at earlier. Yes, it's a cluster of platelets. So good job. So sometimes it may look a little bit like the shapes inside a neutrophil, but there's no cell membranes. That tells you it's not. Excellent job. Y'all are doing great. So this next pair is a little faster to go through. So we've got monocytes and lymphocytes. So these both are a single shape inside of the cell. Monocytes are the ones, just based on their activity, they attack microbes, bacteria, and such, but also work as macrophages. So they eat debris. And then the lymphocytes are the part of our immune system that is fighting. They create the antibodies. They fight off bacterial and viral infections. And they're the ones that step up whenever we get a vaccine to protect us. They learn what to fight for to protect us against infections. And so of these two, again, they both have the singular nuclear shape, but you can see a little bit of difference, especially it's more dramatic in my drawing. So the lymphocyte, it's much rounder, whereas monocyte, it's more irregular. Often it kind of has a bit of a C shape or like a bean shape. You might also notice that the lymphocyte's a lot smaller. It's only a little bit bigger than the red blood cells. And that's the only one of the white blood cells that is smaller and significantly smaller in that way. So that's a big sign that it would be a lymphocyte. And then finally, you notice that like on the left, the monocyte has some vacuoles. That's the little openings. That's part of where it absorbs to do that macrophage, the eating activity. And then also notice how common they are. So lymphocytes are our second most common white blood cell. So 20 to 40% of our white blood cells are lymphocytes, whereas only 2 to 8% are monocytes. So again, when you're looking through, if you're doing our activity, you're going to see a lot more of the neutrophils that we had before and the lymphocytes. Those are majority of what you see. And then you'll see some of the others. So again, we're going to test your knowledge. Which one of these is a lymphocyte? Got A, B, C, D. Y'all did so excellent with the previous one. I expect great results here. So I'm seeing C, I'm seeing B and C. So C's have it. Yes, C is a lymphocyte. So can anyone tell me what B is? It's not a lymphocyte. Look at the shape of it. It's much bigger than the red blood cells, unlike the lymphocyte. Yes, so B is a monocyte. So yeah, it's got a couple of vacuoles. it's not as C-shaped as our example one, but it is still an irregular single shape. Great job. And then, of course, A and D are more neutrophils because they're all over the place. We have tons of those. And then finally, our last lone white blood cell is the basophil. This is the extremely rare one, less than 1%. We don't always see them in our blood smears because they're only around when they're needed. So they're our alert system. They respond if there's foreign substances. They release histamine. They're the ones that give us that lovely itchy response with allergies and asthma, but they're doing their jobs. Just sometimes they overreact. And the way they're shaped is they have kind of a C or S shaped nucleus, but it's almost hidden by the large dark purple granules. So you can see in the image here, and even in my example drawing, like the granules really take up a lot of the coloring, but you can see here on the left side is the nucleus. Again, like I said, very, very rare. You're not going to see them very often. So last chance for you to try out your skills here. You now learned about these 5 types. So what I want you to do is take the first letter of the different white blood cell types and tell me in order what A, B, C, and D are. So just give me 4 letters. See if you can get these. I'll give you a few moments to work your way through it. And it's perfectly fine to guess if you're not sure, because that's how we learn. Because we all make mistakes at the beginning. But the more you do it, the more you'll get correct. Who's going to be our first one to put in their answers? You can try too, Julie, Francelia, Amanda, all of you. Yes, okay. Getting some answers in there. So I see 2 correct so far. So we've got the first one is an eosinophil. It's a little hard because that dark cytoplasm is almost obscuring that, but it's 2 shapes with kind of, it's almost like a pair of glasses that's been a little bent. B is a lymphocyte, C is a monocyte, and D is a neutrophil. So great job for those of you who stepped up and gave it a try. So you're all ready to do our project. So why do we do this? Why do we care? We mentioned we're looking at the health of these animals. And so in the workshop, or the class, when I do this in the classroom, I will also go through with the students. They'll actually classify 50 white blood cells. So here's the answers, like if we had done the full analysis that they had done. And what I like to do is have them look at this and then calculate the percentages, because of course we can't just compare the raw numbers. Once we calculate the percentages and then look and see which ones are any of them out of the normal range for those. So if we look at those, does anyone notice in particular one that particularly stands out? So I'll go ahead and highlight it. It's the eosinophil. Yes, I see. Someone else did step up, couple. So yeah, eosinophils much higher than normal. So what might that mean? So here we've got some information, and this is something that the students have available when they're doing their worksheets, is a little bit of information about what they do and what high levels might indicate. So with this in mind, thinking about this is from a monkey out on this island, what might be going on with this monkey that maybe is making it feel a little bad? Can anyone hazard a guess? Yes, parasite. So that would be a good suspect. Like we may think, okay, this monkey may actually be feeling a little bad because they may have a parasitic infection. That would be an example of why we might see a higher level of eosinophils. So great job. So one other aspect of this project, just to show how it's kind of transitioned through the years, we continue to improve it. And one way has actually been through a collaboration with one of our collaborators at Arizona State University with a group of students that actually used AI, basically a machine learning algorithm, to use image analysis to help us with this project. Now, they set up 3 different models. One was what we call the Goldilocks test. And this is where we take the original image, break it into small pieces, and their model there initially tested whether the spread was good, because you want to look in the areas where the blood smear is well spread. If it's too tight, it's called dense. If it's too sparse, spread out, we call it sparse. And then in the middle, if it's well done, we call it good, which is, why we call it Goldilocks if you're familiar with the children's tale. It's the just right. And so those good cells, images, get moved on to the second workflow, which then looks at them and tries to just find the white blood cells. It doesn't try and type them, it just finds them. So here's an example where 2 are identified in this image, and the number next to it is the confidence that it has. So 87, 89%, excuse me, confidence in that it is a white blood cell. So that is then sent on to the next model, where they tried with the third model to actually classify these cells that were identified. Now, these particular ones, the first one was identified as a lymphocyte, and the second one is a neutrophil with a pretty high confidence, and those were correct. And it did a decent job with those 2 cell types. But remember, those are our most common cell types. We have lots of examples of those. It really had a tough time. It really could not identify the more rare types because we just don't have enough images to train it. So, because this is a question we get a lot. Why don't you just use a computer to do all this? Well, our eyes are better right now. Our brains are better at the pattern recognition and identifying these. So we need your brains to help us with this part. But out of this project did come our single cell workflow, where we used to show larger images and had volunteers click on the cells and then choose what type it was. And so you may have multiple cells in an image. Now we show them a single cell, like what I was showing you earlier, a single cell in the middle of a cropped image, and that makes our workflow much more efficient. So lessons we've learned through the years of working on this project, part of it is just listening to our volunteers. They've given us great comments and feedback, and we answer questions on the site. You can interact with us on there. That allowed us to improve our training, as well as some of our volunteers actually added materials themselves. One volunteered to do some training videos using some of our materials. And then working with these students at ASU allowed us to optimize our workflow to the single cell version of it, which made the project mobile friendly, so you can now more easily do it on your phone. And it simplified our data summarization because we only have 1 cell per image, and it's much easier to summarize the 15 people that have looked at that image into one answer that way. So that's my quick summary, or go through the project part as far as the project itself. But now I'm going to hand it back over to Julie to talk about how this has led to some educational parts.
Awesome. Thanks, Marianne. Well, I'm going to put a link to Monkey Health Explorer. If you want to go there and start participating, please do so. We'd love to see you. But we've been doing this project for multiple years, and it has been really awesome to just see that people are super engaged and excited about it. And so we've had some teachers and some students type in, different notes to say, hey, I'm a teacher. I want to incorporate this in one of my classes. This is really relevant to my curriculum. And so, either at the middle school, high school level, or even college level, maybe I want to bring this into an introductory biology class. And so they're asking, how long this project is going. And this project has been going on for multiple years now. And so we envision continuing it for many more years to come. But it's also really great for students because they kind of see the value in it. And so some students just get skills. Some students, like I said, look at career opportunities. But we've also found that med students and vet students are really getting insight into this and really gaining training experience. So when you go to med school or vet school, you have to learn to read smears. And doing this project, you're going to get much better at that. You'll get training through that and experience doing this. So type in the chat, is anybody interested in med school or vet school? Because then this would be a project that would be really valuable for you. It would give you a head start on figuring out how to read some of these smears and thinking through, what do I need to learn as I get to my next step? Awesome. So anybody who's interested in that, think through finding some opportunities like this, because that'll give you some good training, as well as then providing, scientific expertise to scientists. So on the next slide, I wanted to highlight one thing that was actually unique to us that we didn't realize when we started this project. So we ask people to participate. We give them a little bit of training and we say, can you participate in the project, identify white blood cells? But some people are really well versed. We have medical technologists, like I said, vet techs, med students, and some of the people are like adding in notes and saying, hey, if this example here shows basophilic stippling, and if that were a human patient or if that were, an animal that I was treating, I would think that they had lead poisoning. And so that was one of the things that people pointed out and said, hey, could your monkeys have lead poisoning because we see this basophilic stippling? And in talking to the vets, it didn't sound like that was an option. So we went back to the vets a little bit more and realized, okay, well, basophilic stippling can also mean that there's a parasitic infection. And so more likely some of these monkeys have parasitic infections. Some monkeys, you see all of their red blood cells have this basophilic stippling, or a lot of them. And other monkeys is just really rare. So it's a good way to identify other things in the monkey blood that we weren't initially expecting. And so that project, like working with the volunteers and listening to the volunteers, has really been really rewarding for us as scientists. So the other thing that's really cool is now we're getting some other people who want to expand this project. So not just look at monkeys, but look at, say, reptiles from Galapagos. So here's an example of a marine iguana, and we now have a collaborator. So Greg Lubart at NC State and Dan Dabrowski, who's the vet at the museum, collaborate a lot on some of these projects. And so they give us some of these blood smears of marine iguanas from Galapagos and turtles from Galapagos. And we're now starting this project and expanding into looking at reptiles. And reptile white blood cells look somewhat different than humans. So they have nucleated red blood cells, and so their white blood cells and red blood cells look a little bit different. Overall, their blood is different. And so it's giving us a run for our money because we're, creating new training materials for some of these. But I think it's going to be a really valuable project and also really great for training some med students and vet students because they're getting more experience with very unique populations of animals. So if you want to participate in the reptile piece, we have it right now listed on the Monkey Health Explorer site, but as a test for reptile blood cells, and hopefully this will become its own project in the future. So I also want to just tell you a little bit more about some of our outreach in Puerto Rico and North Carolina. So Francelia here has been taking this across schools in Puerto Rico and as well as some public engagement events. So here's a great example of her talking to students and the public about this project and explaining a little bit more about the monkeys on Cayo Santiago, which is an island, where they live. And so it's really valuable to talk to the public and really explain to them, hey, this is a valuable resource. Maybe you didn't know about it, but here's something that's really valuable and is providing some career and job opportunities. And Marianne here is a picture of her at this siren event where teachers come and look for more material for their curriculum, for their schools. And so really great opportunities to connect with the public as well as teachers and students. Francelia, I don't know if you want to say anything or if you have any thoughts, you're welcome to chime in if you want to add any thoughts to this. But I know I'm putting you on the spot, so you don't have to if you don't want to. All right. All right, so, and then I just kind of want to close. If anybody is an educator or works with educators, we currently have some curriculum materials. So we have lesson plans. They are in English and Spanish. We have some activities like this crossword, and we've done some teacher training workshops where we give away these kits. So if you are a teacher or you know a teacher who might want one of these kits, it has the lesson plan and some of the candy to make blood smears. We also have, I don't know how well it'll show up, but Marianne has 3D printed a lot of these cells, so you can take those to your schools or use them as examples of like, oh, hey, here's a neutrophil and here's a, whatever other type of white blood cell you have. So good examples that you can take with you and then play with those as well. They're fun. All right. So with that, I think I'm going to close by just on this next slide, just talking about how you guys can get involved. So again, if you want to participate in the project, please join the project. And Marianne, if you can go to the next slide. You guys can all participate in the project at monkeyhealthexplorer.org. We have a grant in progress that we hope will get funded that'll fund more of the research aspects. So we might have some internships available. We're always looking for research collaborators. We're also looking for some funding opportunities to fund more of that outreach and get into the schools, because I think that's going to be really valuable to get students engaged early and really thinking about STEM careers early. So if you're an educator, we would love to have you work with us, help us refine some of the curriculum and lesson plans, but we also have volunteer opportunities. So please, please reach out if you have any questions or thoughts. Here's email for both of us. I can put our emails in the chat as well. And I think on the next slide is just our thank you takeaway. Yeah, so thank you to everyone who has participated. Like I said, it's over 20,000 people, great volunteers, a lot of awesome collaborators. This doesn't include everybody, but just highlighted some of the people we talked about today, as well as some of our funding. And so thanks everybody for your time here, and check the chat because we have some CLE credit and some other codes that you might want to connect with and find out about this event more. But connect with us if you're interested, and we're open for questions. So please feel free to drop in chat, or you can unmute and ask us questions. Right. Is anybody going to start participating in this project? Who wants to start using Zooniverse and do pattern matching? Hina, yeah, you were good at this. Keep that up. Awesome. Well, thank you all for coming. Again, please reach out. We're here to answer questions if you have any, and we'd love to hear from you and see you on the project.
Thanks so much, guys.
Thank you all. Welcome. We hope to see you. And I will stop our recording.